Hello, my name is Dr. Adams, and I am your professor. Today, we are going to learn about genetics. Now, I know that this is a 5-week summer class, and you probably wish you were on your way to the beach right now. Cruising down the highway in your red convertible with your hair blowing in the wind. You arrive at the beach, and it is absolutely beautiful outside. I mean, the weather is perfect. This is what life is all about. Maybe you'll start your day with a little bit of yoga on the beach. Or maybe you'll go for a swim in the ocean to cool off. Either way, it's going to be an amazing day. Unfortunately, this is going to be your home for the next few weeks, but I promise it is going to be worth your while. So let's get started. What is a mutation? A mutation is a change in genetic material. Mutations can be beneficial, harmful, or neutral. Today, we are going to talk about point mutations and frameshift mutations. When you think of point mutations, think substitution. A point mutation is the substitution of one base pair for another. This can lead to the substitution of one amino acid for another. There are two types of point mutations, transition and transversion. If you look to the left here, a transition means a change from a purine to purine or pyrimidine to pyrimidine. On the right, a transversion is a change from purine to pyrimidine or pyrimidine to purine. When you think of frameshift mutations, think insertion or deletion. A frameshift mutation is the insertion or deletion of one or more base pairs. This alters the reading frame, changing several amino acids in the protein. Let's look at the example on the left. Here, a simple sentence displays the effects of insertions and deletions. If you delete or insert the letter E, the words are no longer spelled correctly and the sentence is no longer comprehensible. Now, let's look at the example on the right. Here we have a DNA sequence. During translation, each 3-letter sequence of mRNA nucleotides corresponds to a specific amino acid or stop codon. If you delete or insert a nucleotide, the entire DNA sequence following the mutation will be read incorrectly. Now we are going to discuss mutations at the protein level. There are 4 types: silent, missense, nonsense, and readthrough. Silent is a mutation with no change in the amino acid. Missense is a mutation that changes the codon for one amino acid to that of another. Nonsense is a mutation that changes the codon for an amino acid to a stop codon. Readthrough is a mutation that changes a stop codon to one encoding an amino acid. Let's look at the example below. First, we go from glycine to glycine. Because the mutation codes for the same amino acid, it is a silent mutation. Next, we go from lysine to isoleucine. Because there is a change in the amino acid, this is a missense mutation. We then go from tyrosine to a stop codon. This is a nonsense mutation. Last but not least, we go from a stop codon to leucine. This is a read-through mutation. This is a flowchart to help you figure out the type of mutation at the DNA level and protein level. First, you should ask yourself, is it a substitution, insertion, or deletion? If it is an insertion or deletion, it is a frameshift mutation. If it is a substitution, it is a point mutation. Next, determine if it changed from purine to purine or pyrimidine to pyrimidine. Or did it change from purine to pyrimidine or pyrimidine to purine? This will determine if it is a transition or transversion. Then, at the protein level, it will either be silent, missense, nonsense, or read-through. Now, let's look at a practice problem. First, is this a point mutation or frameshift mutation? At the DNA level, this is a point mutation because the normal strand has cytosine and the mutant strand has adenine. All the other bases are the same. Is this a transition or transversion? We went from cytosine, which is a pyrimidine, to adenine, which is a purine. At the DNA level, this is a transversion. Next, what kind of mutation is this at the protein level? Let's look at our mRNA strands first. Remember that AUG is the start codon, and UAA, UAG, and UGA are the stop codons. Also, please note that DNA contains thymine and RNA contains uracil. Here is a codon chart for translation. I'll walk through the normal strand for practice. AUG codes for methionine, AGC codes for serine, GUA codes for valine, CCC codes for proline, UAC codes for tyrosine. Then there is a stop codon. Based on the normal and mutant strand, the amino acid proline changed to threonine. At the protein level, this is a missense mutation. Let's walk through one more practice problem. Is this a point mutation or frameshift mutation? At the DNA level, this is a frameshift mutation because the normal strand has GTA, and in the mutant strand, we can see that TA was deleted from the sequence. Therefore, this is a deletion. Now, let's skip over to the amino acids. Here we see that the stop codon was changed to the amino acid lysine. At the protein level, this is a read-through mutation. All right, that concludes the lesson for today. We are off to a great start. See you next class.